And we're live apparently. Um, this is the sort of next episode in this little series of looking at published papers and dissecting them. It just so happens that the first two that we've tackled were looking at problems with the papers rather than just explaining the papers. So tonight, the paper that we're having a look at is one entitled "The Evolutionary Accessibility of New Ends." Oh, I've done a JD, Kane. I've done a JD. My own voice just interrupted me. Right, anyway, it's become an internet term, that, hasn't it? I've done a JD, Ken. Right, anyway, the paper is Evolutionary Accessibility of New Enzyme Functions, a case study from the biotin pathway by Gauger and Axe. Now, it's Anne Gauger, whatever, and Doug Axe from the Discovery Institute. Paper was published in 2011 in the in-house journal of the Discovery Institute, and that's called Biocomplexity.
Sorry, in-house journal.
Yes, in-house journal.
That makes it really useful then, doesn't it? If it's an
yeah.
journal.
This paper is one of the ones that gets touted as a peer-reviewed support of intelligent design or evidence for intelligent design. But there is a big problem because it's in-house, and if you look at the editorial board, they are all intelligent design advocates, C-design proponentsists. I think there is one botanist there. I'd need to look at it again, and I think that might be an interesting hangout as well, having a look at the background of the editorial board. Now, it's the editorial board that are the peers that do the reviewing.
This is like, it's like giving yourself a high five, isn't it?
Yeah. So
basically, it's like clapping.
Yeah. It's not a case of, usually with a journal, when an editor gets in an article, and he looks at the actual paper itself, and he then chooses referees from that field. Now, he's not just looking at people associated with the journal. He's looking out at all the people in that field of research. I mean, I used to get in papers to review. My husband, he's still working in the field. He gets papers in to review still. And all of that is done free of charge. You don't get paid to do it. Now, with the Biocomplexity Journal, this is all kept in-house. It doesn't go out to external reviewers to find the problems. So anyway, this paper has been one of the ones touted as providing evidence for intelligent design. And I mean, on the face of it, it looks fine. But there's a really bad design problem with it.
It is a great looking paper. It's beautiful.
Yeah. And when you read the abstract, Cliff, I think the link's in the side chat. If you want to link to the biocomplexity, it was just after you joined. You'll see the link.
Yeah, let me. I'm just waiting for it to load up.
And if you just screen share the abstract, I mean, it sounds nice and sciencey.
1 second. Trying to zoom in. Oh, the side is zooming in. You zoom in with.
The typesetting is great. The figures are great. Looking like the actual images they use are. Boy, it looks so sciencey. And
I'm trying to present Cliff to all, and feeling. I think, is that you presenting to all, Cliff?
No. Do you want to share controls with me? I should be able to.
Have we got present to everyone?
Yeah,
right. Yeah. Right. So if we look at that, we can see it looks really quite good. Enzyme group Enzymes group naturally into families according to similarity of sequence, structure, and underlying mechanisms. Enzymes belonging to the same family are considered to be homologs, the product of evolutionary divergence, whereby the first family member provided a starting point for conversions to new but related functions. In fact, despite the similarities, these families can include remarkable functional diversity. Here we focus not on minor functional variations within families, but rather on innovations, transitions to genuinely new catalytic functions. Prior experimental attempts to reproduce such transitions have typically found that many mutational changes are needed to achieve even weak functional conversion, which raises the question of their evolutionary feasibility. To further investigate this, we examined the members of a large enzyme superfamily, the PLP-dependent transferases, to find a pair with distinct reaction chemistries and high structural similarity. We then set out to convert one of these enzymes, 2-amino-3-ketobutyrate-CoA ligase, to perform the metabolic function of the other, 8-amino-7-oxynonoanoate synthase. That is a word I'm struggling with. After identifying and testing 29 amino acid changes, we found 3 groups of active site positions and 1 single position where KBL2 side chains are incompatible with the BioF2 function. Converting these side chains in KBL2 makes the residues in the active site cavity identical to those of BioF2, but nonetheless fails to produce detectable BioF2-like function in vivo. We infer from the mutant examination that successful functional conversion would in this case require 7 or more nucleotide substitutions, but evolutionary innovations requiring that many changes would be extraordinarily rare, becoming probable only on timescales much longer than the age of life on Earth. Considering that KBL2 and BioF2 are judged to be close homologs by the usual similarity measures, this result and others like it challenge the conventional practice of inferring from similarity alone that transitions to new functions occurred by Darwinian evolution. Sounds great.
I was with you all the way up to abstract.
Yeah. But it does. That sounds absolutely fine. Until you think about what they're trying to do. They are trying to take one enzyme and turn it in to another enzyme. And there's the problem right there. If you remember, we do not say that humans evolved from chimps. We say that humans share a common ancestor with chimps. And there's a huge big difference there. Massive difference. Now, I'm going to stop that. In fact, I'm going to keep presenting to all, or have I been and gone and stopped it? Cliff's muted.
Yeah. Cliff, you're muted. I think Cliff stopped the screen share there. We're back.
Sorry, I stopped the screen share because I didn't think you needed it anymore.
Yeah, right. And he's still presenting.
Now, what I would like Cliff to do is I sent him a wee scabby diagram earlier on. If you could screen share that scabby diagram. That's the one. Okay. Now, in this diagram, we have A. That's our common ancestor. Okay? And we will call B and C the 2 enzymes. And they're trying to turn enzyme B into enzyme C. And here's your problem. How would you turn B into C?
Yeah, it's a bit difficult if they've gone off a different route, off a different branch.
You would have to go all the way down the branch from B to A, and then all the way back up from A to C. Now, the claim for these 2 enzymes is that they had a common ancestor and that they diverged off. And what they're trying to do in the lab is to actually get double the number of changes that would be required, because they've got to undo all of the changes from B to A, and then put in all of the changes from A to C. So they're actually trying in the laboratory to force these proteins to take a pathway that nobody has ever claimed they took.
Yeah, I can see this as being a problem.
Yeah. Now, if you look at this very carefully, and we think about this as a family tree. Now, I'll say this at this point here. Oh, wooga wooga! Analogy alert! Analogy alert! A, okay, is grandparent or grandparents, right? They have two offspring, and the two offspring then have two offspring. B and D for one of them, and E and C for the other. Okay, so they are now the grandchildren of A. So that makes B, D cousins of E and C. Are we following this so far?
Yep.
B and D would be siblings, and E and C would be siblings, but they would be cousins to each other. Okay? And what they're trying to do is they are trying to turn one cousin, B, into his cousin C.
Yeah, well, they're trying to turn cousins into siblings.
No, they're not, because they're trying to turn them. They're trying to turn one enzyme into the other enzyme. So they're trying to turn cousin B into cousin C.
Right. Yeah.
As if one descends from the other, so that they're saying that.
And
So
what they're saying is
pathway from my grandfather down to me.
Mm-hmm.
And, they're saying, well, let's find the hereditary pathway from me to my cousin.
Mm-hmm.
Right? How plausibly could my cousin have descended from me?
Yeah, and that's the problem, because they are saying that because they can't turn enzyme B into enzyme C, that B and C cannot share a common ancestor, which is the same as saying since B is C's cousin, and because they can't turn B into C, they cannot share a grandparent. But they obviously do.
Yeah, makes sense.
And there may or may not be a pathway to converting B to C. That may well, obviously there is, like, because these are chemical transformations, so you can actually go back up the chain. If there's a way to convert B to A, you could convert A to C. But there may be another pathway. I'm just sort of picking up, Fiona, when you said a moment ago about how, to go from B to C, you have to go through A.
Yeah.
And I wonder if you, well, I wouldn't say you have to. I don't know that that's necessarily the case. There may be other pathways.
Mm-hmm. Yeah.
There may be similar amino acid sequences that would there might be a different pathway to go from B to C. But in any case, no one's proposed that pathway. The proposed pathway is from A, which is a more generic enzyme, to you have to unlabeled, where the forks in the road are there, to two other sort of more specialized versions of A. A prime and A double prime, whatever you want to call them, and then A prime further specializes into B and to D. That's and then of course the other one further specializes into E and to C. That's the proposed pathway that's unchallenged. And I mean, there may or may not be a pathway from B to C.
Yeah, and see, the difficulty here is that this evolutionary pathway for the enzymes, nobody knows what A is. So nobody knows what the starting...
That's a problem for everybody, yeah. Sorry.
Yeah, so if nobody knows what the ancestral enzyme was, then they don't know how they've gotten to B and to C.
Although, don't you think that's something that could be worked out? Like, as there are obviously some sequences that are invariant amongst all four B, D, B, C, D, and E. There's going to be some sequences that are invariant, and some that are variant but common between B and D, and some that are variant but common between E and C. Like, surely you should be able to work out large stretches of what A must have been. to
Yes,
yield.
yes, you can. I mean, for the time being, ignore D and E because they only worked with two enzymes, B and C.
Yeah.
And
Well,
just put the
yeah, then they have no starting ground whatsoever. The fewer that you have.
exactly.
I mean, when I'm trying to, I mean, sometimes these things are difficult to discuss because the information we get, you know, for whether it's enzymes or DNA sequences, or for whole species, when you look at morphology of whole species, you can't just sit down and say, well, here's an A and a B and a C and a D, right? Like, I mean, obviously this is meant to be sort of schematic, that these things are thousands or millions or trillions of things we're talking about, whether they're enzymes or sort of in general proteins, or amino acid sequences, or even the morphology of whole creatures. So they only so if you, it's like this effort we've had, like you take blood samples from all the mammals, and you can come up with the tree of life, you know, based on DNA sequencing. It's not really that informative if you only did it with four mammals, right? It's
Yeah,
really informative and compelling when
yeah.
they've done it with tens of thousands of species. So
And
if they're only talking about B and C, well, then they have no foundation at all to be
yes,
talking about what common
yeah,
his historical basal enzyme
yeah, and
that would have led to B and C.
yeah, and basically they don't even mention that the historical basal enzyme. All they've done is show that they cannot change B into C. They have taken B, they've made some changes to it, and it didn't become C. So they've, in effect, taken one cousin, made some changes, and it didn't become the other cousin.
Yeah, and this is the kind of thing, like we were kind of wrapping up the last episode of talking about the role of peer review and trying to disavow people of the idea that they don't analyze the data. When you submit a paper for publication, they don't go over all of your actual data. Although I do have a question about their procedures, but in any case, just to make this point, they don't actually go over all your data and make sure that you analyzed it properly and that all your charts are good. They look at the general form of the paper and say, is the methodology appropriate for this sort of an investigation? Do the exhibits presented properly capture the types of results you would get from this methodology? And then most importantly, do the results and conclusions, or do the conclusions follow from the results as described? And this is something that I just cannot see this paper passing peer review on that last ground, right? And I'll admit to, I was reading the abstract and reading along with you as you read it, and I have to admit to the confusion. I don't even know what compounds they're talking about. I mean, I would have those three sentences in the middle. I would have a lot of trouble reading without tripping over my own tongue and being unsure how certain chemicals are pronounced. But at the end of it, when they where is the thing here? There's just no way that observation. Evolutionary innovations requiring that many changes, or is it here, would be extraordinarily rare, becoming probable only on timescales much longer than the age of life on Earth. Well, no, it just all of a sudden there's a probability thing that comes from their inability to do something, and they jump to a timescale all of a sudden. Like, where does that come from?
Yeah, and what they've done is they've set out to try and debunk the idea of a common ancestor for the two enzymes, the two homologs. But the experiment that they set up looks at attempting to change one into the other without going back down to the common ancestor first. They've tried to go across the top of the diagram.
And there may or may not be such a pathway. I wouldn't think there would be, and there's a reason why I think that. You probably know that reason better than I do, and I do want to touch on that. But I got a question. It's such a dumb question. When we say, they were trying to convert this to that, whether it's B to C or whatever, how are they trying to do this? And this is dumb. Like, most labs, I would say, presumably there's people in white coats and they've got beakers and Erlenmeyer flasks and Bunsen burners and whatnot. Is this actually their laboratory research?
Yes. What they did was they changed. It says in the paper. It says After identifying and testing 29 amino acid changes, we found 3 groups of active site positions and 1 single position where the KBL2 side chains are incompatible with BioF2 function.
Mm-hmm.
So they found 3 groups of active site positions and a single position where they can't convert it.
Right, but I'm wondering when they say they
And they're
found
talking
these.
about the active site of the enzyme. That is one of the most sensitive parts of an enzyme, the active site. Mutations in the active site can be pretty catastrophic. Whereas a mutation in the more structural part of the enzyme may result in a slightly less efficient protein. It may result in one with same efficiency. So what they actually did was they went in and they changed the amino acid sequence of the protein, and it didn't work. They went in and they changed the active site, and it didn't work. It no longer.
Well,
it didn't become the other one, shall we say?
Does that not happen anyway?
Well, yeah. I mean, what they did was they changed the active site of B, and it didn't become C. That is what that bit's saying there.
I mean, I know this sounds stupid. I mean, I don't, because obviously I'm no good at biology. I don't understand these things. But when you hear the creationists talk, they sort of say, you know, monkeys turned into man. That's what you believe, monkeys turned into man. And I'm wondering if they're looking at the same point. They're sort of saying one thing changes to another.
No. Because the chances are the way that they've done this is by changing the DNA sequence itself.
Yeah, but that's what I'm wondering. How did they do that? Like, I'm just trying to find an example, thing from the description of their methods.
You actually need to go down to the paper to have a look at this. Let's look at the methods.
Yeah, I see. Approach, and it's got stages of an approach.
Yeah, approach, and that.
Where do you want me to go?
Right. Okay. Scroll down the paper, and you'll see a wee colourful diagram.
Yeah.
I mean, there's photographs. Figure 13 shows photographs of growth medium, which is actually not a helpful figure.
No, but that one there with the colours on it, right? Look very, very carefully at the title of the figure. Cliff, you're echoing.
Oh. Is this figure 1 you're talking about?
Yep.
Yeah. A structure space mapping of the evolutionary history of a hypothetical enzyme family.
Hypothetical.
Yeah. So. Yeah, and they don't. Okay, under what hypothesis? Like this is a prediction that follows from what hypothetical or theoretical account. I don't know.
Yeah.
It's a doodle. It's a doodle on the page, which may be helpful to explain what it is, what the what the principles are dealing with. But everything I've seen in here, they've got this Q parameter defined in terms of a recombination of the root mean square deviation. They've got their little they got these formulas in here, but I don't see, did they actually manipulate chemicals in this thing? There's no description of the technology, and I may have missed this, maybe my inexperience with this level of chemistry, but I don't see where they say they did the laboratory work. Do you see what I mean?
Right, that's what I'm looking for now.
BioF sequences for the first method were found by performing a blast search on the concise microbial protein database.
Hold on.
Okay, that's a database query.
Where are you on that?
I'm sorry, I'm zipping up and down rather cruelly. I'm on top of page 7. That's actually the first words of page 7, which is included in their stage 1 description.
Okay, right. Right, okay. So they constructed a mutant BioF gene, it says. Plasmids carrying these mutant genes were introduced into an engineered strain of E. coli lacking the chromosomal BioF gene. So you've got an E. coli that's got is missing the BioF gene in its chromosome, but you're able to introduce it on a plasmid, which is a small circular piece of DNA that you can actually put into bacteria, right? And if you put that missing gene onto the plasmid and then transform the plasmid into the bacterium is then able to use the BioF gene on the plasmid to make up for the fact that it doesn't have it on the chromosome. And what they're looking for is what they're calling is biotin auxotrophy. Now, that means, can the organism manage to grow without biotin being added to the growth medium? I.e., can it make its own biotin? And the genes that they've been looking at, these enzymes they're looking at, are steps in the biotin synthesis pathway. So if you've got a version that doesn't work, then the organism will not grow on a medium lacking biotin. You would have to supply it with biotin because it can't make its own. You following?
Yeah. Oh, yeah. And I buy their description. And most people just remember this, but that test using the biotin pathway is really nice because it's very sensitive. Because they don't, if I understand this correctly, they don't. E. coli don't need a lot of biotin in their diet. They need a way of processing the little bit they have, but it's considered very sensitive because if they don't have a way of processing it at all, they're doomed if they're not provided. And if they do have a way, well, then they're I screwed that up. That description, I got it backwards. The point is, it's what they call it, like a statistically sensitive thing. You're going to observe a really big effect. Either this enzyme is effective or it's not effective, and you're going to be able to tell. It's not going to be a fuzzy thing when you're looking at these petri dishes because they actually don't need a lot of biotin to continue functioning.
Yeah, and the other thing that's interesting is it says in that first paragraph of stage 2, interestingly, of these 15 single amino acid substitutions, the only one disruptive enough to produce the bio minus phenotype is the replacement of histidine 152 with asparagine. Now, that's a change of an amino acid, and yet it doesn't actually disrupt it. And now, to change the amino acid, obviously, you've got to change the codon. That's 3 bases. And yet we have heard the argument from, I think it's Nephilim 3, that single nucleotide polymorphisms are always detrimental.
Deadly,
and yet,
deadly, he'll tell you. Always.
Yeah. And yet, here we have only one of the 15 here that is a complete amino acid change. Made no difference.
Sorry, amino acid change or single nucleotide change?
Well, that's the thing.
You're right. You're right. It was amino acid. Sorry, you're right.
Yeah, it's a whole amino acid change, and yet the argument has been made that a single nucleotide change is always disruptive. But a single nucleotide change can result in the same amino acid. So that's even less chance of it causing a difference. So we've got only one of those was enough to produce the bio minus phenotype. I just think that's really quite interesting. But if you squiggle back up to the colour diagram, that round one that we had, and look at it carefully, do you see the little sort of greyish blob in the middle? Sort of beige thing?
Yeah,
that's the equivalent of A in my diagram. And then the pale blue, the dark blue, the red and the lilac are the equivalents of B, C, D and E. I've just made it a straighter pathway. I used the letter Y to show it.
Yeah, well, they certainly don't want to make a simple diagram that shows, here's the beige one, and then for some reason it trifurcates into 3 different functions.
Mm-hmm. And remember, this is a hypothetical enzyme family.
Oh yeah,
that's the actual family of enzymes that they are examining. Which leads me to think to myself, why bother with a diagram? And if you look at the figure on page 4.
Figure 2 at the bottom of page 4, yeah.
Yes. Now that is a near neighbour structural distance graph for all of these enzymes that we're talking, all these homologs that we're talking about, that have all these different functions.
Yeah. Now this is analytic. I wonder, probably worth explaining. I don't know how well it comes across on the screen, but this isn't just a hypothetical. Well, let's doodle this out to show the relationships. On each of the lines, so there's solid lines and there's dashed lines, and on each of the line is actually a number. Calculated to 2 decimal places, and that number is actually an indication of, I was going to say strength of relationship. It's actually the opposite. It is the distance. When they say nearest neighbor, there's actually a metric to calculate, well, how similar are these things, or actually really the number is more of a measure of dissimilarity. So when they're physically close together on the page, that means that the sequences are quite similar. When they're further apart, there's going to be a bigger number on that line, and it means that there are numerous differences in the sequences between them. Now, the actual placement on the page is a little bit arbitrary. I mean, they physically put them on the page so that the distances match the numbers, but like if you look off that little wing on the left-hand side, those little golden ones, there's that one y, was it 1WY, 1WYU colon D, and 1W, yeah, those two over there. That I mean, why does it go off at like 11 o'clock, right? That's an arbitrary angle. It just makes it fit on the page. But the distances.
It may be because it comes closer to the one JS6 above.
But there's no metric there, right? Like they'll. My understanding is that they would show that as being, you're sort of more constrained when you have a similarity metric between that one and the one JS6.
Yeah.
But in cases where it's just like, well, we've got these pairwise comparisons and we don't have a metric of similarity between those two, well, then they just sort of stick out like sticks all over the place.
What is the difference between solid lines and dashed lines?
That is what I'm actually trying to find out, to be honest.
I would have to dig through my memory. I wonder if it's in the description on the...
That's what I'm looking for.
Yeah, there's the thing I was saying. Layout and distances between unjoined nodes are arbitrary.
Yeah, dashed edges connecting enzymes having different chemistries. Grey edges radiating from nodes with poorly characterized functions, and backshaded edge, showing the functional transition examined here.
Or that one gray edge in there that shows the transition. Okay.
Yeah.
Yeah. No, I mean, like, say these.
I don't think there's only one, because if you look at the green bit up here, that seems to have a lot of gray in it.
No, I mean there's the sort of chubby gray part over by 1FC4 and 1DJ9. That's sort of like cloud highlighting to say this is what we're talking about here.
Yeah.
But yeah, they do have gray and then black and then dashed. And you know, in any nearest neighbor analysis, you can assign different meanings to those things. I just don't know what they mean in the context of this paper. Backshaded edge.
Yeah, that do they just mean it's got a black highlight on it, so you can actually see that as a thicker line?
Yeah, yeah. But yeah, obviously, the thing is, I don't know what this term means. Like this gray edges radiating from nodes. So the gray edges refer to nodes with poorly characterized functions. So I have to know what a poorly characterized function is to understand their choice for doing so. But
and the dashed edges connecting enzymes of different chemistries. But why are they connecting?
Yeah. Okay, sir, I hope that didn't derail. I just wanted to make the point that since we were sort of scoffing at their little doodle from Figure 1, that this doodle is actually established from it comes from a well-established statistical analysis technique where the numbers on there are actually meaningful things, and you want to take a lot of meaning away from the lengths of these lines, but not much meaning away from the angles that they come off at. So if it looks like there's a cluster, it's only a cluster, and also ignore the colors. That's a totally arbitrary choice on their part.
Yeah.
That when you see a cluster, they need to be physically close together, not just coincidentally near another branch. That doesn't mean anything. And I apologize if I just wanted to make sure that wasn't mistaken with Figure 1.
Yeah. No, that's perfectly true. But But it shows how complex this family is.
Sorry to be late, guys. Work ran a little late, and then I had to make dinner.
We've got ourselves. We are looking at the. Have you seen the Gauger and Axe paper?
And Gauger, yeah.
Gauger, is that how you say it?
Yeah. Well, that's how I've heard it pronounced.
Right, that'll do. Gauger. That's what I used to call
Told you the other day, didn't I?
I've met Doug Axe. I ran into him in Texas, heard him talk, asked him a question on this topic, in fact.
Oh, yeah.
Yeah, I actually made a video on this one, if I
That's
recall
why I
correctly.
wanted you in on it. Yeah, because it's so interesting. We've gone through the abstract and we've been messing about with a diagram of mine, trying to demonstrate what the cousin's problem is and why they've set out to answer the question of could these have come from a common ancestor, but they design an experiment that doesn't do that. It asks, can we turn one cousin into another cousin?
Exactly. Yeah, that's exactly right. And they pick one that was already broken, right? So they're testing their forward and reverse mutation rates.
Yes.
So they've taken one that's already 2 steps broken.
Yes. And the problem with that, I think, is that you can't set out to answer the question, do these people share a grandparent? And then design an experiment which actually answers a completely different question, and then come to the conclusion that therefore they don't share a grandparent. The design of the experiment does not answer the question that they think they're asking.
So I had a moment. I want to describe this moment to you when I'm attending this intelligent design presentation.
Was this Anne Gage's presentation?
Was that?
Was that the presentation that she gave?
No, Anne wasn't there. It was
Okay.
Doug Axe and what's his name? Von whatever. I'm blanking on the name. It was a few of the major ones. Doug Axe is the only one at the Discovery Institute with, and he's not at the Discovery Institute. He's at Biologic Institute, which is a wholly owned subsidiary of Discovery Institute.
Yeah.
He's the only one I would say has legitimate scientific credentials that I respect. And Gauger, maybe, yeah, kind of a third-tier kind of scientist. But Doug Axe had a good background. But there was a moment where they were answering my question where they suddenly backtracked. So he gave the correct answer. The question was about whether or not novel functions can come from sort of junk DNA, whether or not neofunctionalization is a real thing, is basically what I wanted them to admit to, because they're talking about, mutations can't do this and mutations can't do that, and I said, mutations aren't really what we're talking about when we're talking about novel functions. We're talking about family splitting. We're talking about divergence of sequence, which can produce novel functions from already working models. And we talked a little bit in my question about the shape of evolutionary space that can be accessed. And he gave the correct answer. And then he said, but, we don't observe that in nature. Or I don't think we've been convinced of this. In other words, he responded like a scientist, realized his mistake, and then answered like an ideologue whose salary depends on being an ideologue. And that's the point where I realized that these are not people who necessarily, in my opinion, genuinely hold these beliefs, or if they do, they've got this compartmentalization in their mind where they can sort of do both in their head. Which for me it was a revelation. I had assumed that these were people who were ignorant, and now and I've often said not to do this, but now I assume they're malicious, that they're intentionally deceptive, and I tell people not to do that because it's the wrong thing to assume about people who disagree with you aren't necessarily ignorant or deceptive. They could simply have a different perspective. But when he made that mistake, that's when I decided that, and I'm thinking of Sternberg, Richard Sternberg. But Doug Axe was in on the question as well. So have we talked about evolutionary search space or fitness diagrams?
I was hoping you would come in at that point, because that's more your field than mine.
So this would be great if I had a visual for you.
Well, we've got now let me think. We've got a near neighbour structural distance graph.
Structural distance graph.
Yeah, page 4. Cliff, could you reshare that horribly convoluted diagram, please? Figure 2.
Yeah, I'm just trying to think of what would be a quick source for that. I mean, figure 2 from it shows this the near neighbor analysis based on the sequencing done by others, I believe. Although I'm guessing, if I'm thinking of the right video that Concordance is talking about, you're talking about a fitness assessment that goes with this too, like exploring an evolutionary space.
That's right.
Yeah, you might have to
guide us. Which is more conceptual than actual. Mm-hmm. But you know, another one that maybe everyone might be more familiar with is Dawkins talks about, you know, Mount Improbable.
Mount Improbable, yeah. Do you want to guide? Which you might, just as a suggestion, can you think of an exhibit that would be helpful, and then we can kind of search for it while you're.
I put it in the description. I've got my video pulled.
Excellent.
Oh, okay. There we go.
Yeah. Right, Cliff.
So Cliff's got that, yeah.
Yeah, and go to 207 in the video.
This might not be the paper. I'm actually double check what paper I was reading when I came up with this, but it's the same argument he makes every time. Yeah. He really is a kind of a one-trick pony when it comes to intelligent design arguments.
I'm going to just wait for Cliff to find that.
Sure. But Mount Improbable, the idea is that there are two faces on this giant mountain. You've got the sheer face, you know, the one that no one could possibly climb. And if you start from that side of the mountain, you say this is an unclimbable mountain. It cannot be climbed. But if you step around, and this is what really biology does, what genetics, nature do, is it explores the space at the sort of base of this Mount Improbable, and it finds the other side, where there's a much broader approach, and the slope is a lot shallower. And what they've intentionally done is started from a broken protein, which would have only one way of achieving functionality in the biotin pathway, and that's through recreating the specific events that undo the damage they did. In other words, one very narrow path up the mountain, as opposed to these other paths from the other side of the mountain. And here you've got Doug's model on the left, which says basically only one possible outcome gives me any kind of function, as opposed to the reality in nature, which is I'm not looking for a biotin pathway. I'm an organism, and I'm trying to metabolize things any way that I can. I'm trying to eat food from my environment, whether that's the grass or my fellow bacteria, whatever it may be. I'm exploring the totality of all possible strategies. And what Doug is really doing is saying, if we break this in a very specific way, can it retrace the steps back up that exact set of events? Mm-hmm. And that's not the real scenario. What he's done is verify an old principle, not really well supported, but we won't go into that, called Dollo's Law. Do you guys know Dollo's Law?
No.
It's named for a Belgian scientist a long time ago. Don't ask me details. The idea is that nature never recapitulates itself. It never recreates birds from organisms that have evolved from birds, right? We'll never have a dinosaur evolve back from a bird. You might say that they're one-way paths along evolutionary parameters, and that's because there's only one way that you could ever go back to an ancestral form, and that's through the specific set of events that got you to where you were.
Exactly, and that's the point I was making when I said that basically what that they're trying to do is turn B into C. When it shares an ancestor A, they would have to go all the way from B down to A, and then A back up to C.
Yeah, exactly.
That was the diagram. Yeah, and whereas their experiment was trying to bridge the gap across the top without going down to the common ancestor.
There's that. article
Yeah,
in.
that was my diagram I threw together earlier on today.
No,
So
that
yeah,
illustrates it very well. Yeah.
yeah, I'm just going to present that to everybody. Yeah, so just recapitulate what you said there about that, about having to go all the way down.
Yeah, exactly. So, A can go to B, D, E, or C, and those are all acceptable, favorable outcomes that increase the fitness of the organism. But the specific path to go from B back to A and then up to C is highly improbable. So retracing back from D to A through these specific chain, only this one base out of 3 billion bases or out of 3 million bases, the chances of that one base being the one that mutates, yeah, that's a very low probability event. It's a little bit like saying you can never beat 3 jacks because the only way you could beat it would be to deal 4 jacks in this specific order. Well, no, there are a lot of cards that will beat 3 jacks. And if you ignore everything but that one particular hand of cards, yes, in fact, you could never beat 3 jacks with 4 jacks because the chance of dealing 4 jacks is very, very low on the very next deal of the card.
And I think the way they designed their experiment, though, they weren't trying to go from B to A to C. They were trying to go from B to C.
Right.
Across the top.
Yep. Yep.
But the question they were asking was, could they go from B to A to C? But the experimental design asked the question, can we go from B to C across the top? And they then used the answer that they got from that experimental design to then answer the question that the experimental design didn't ask in the first place, which they thought they were asking. So the last one question.
Can you turn your cousin into your cousin? Or can you be turned into your cousin? No.
I have a question. When I studied psychology, one of the things that came up with was ROG. If you're going to have an experiment, it has to be replicatable, objective, and controlled. Do you think they wrote this knowing that it wasn't possible anyway? They already knew the outcome, so they wrote it knowing it wouldn't work.
Can I share a story about Anne Gauger? This is, I think, a little funny.
Is this the Wistar thing?
Yes, yeah. You tell it.
Don't worry, I'm coming to that one because I've got a screen share ready for that one.
I'll hang off on it. I think that's hilarious, though.
Yeah.
That's another example, though, where there's a duality of, yes, I'm a scientist, but also I'm an ideologue, and my job is about the second half of that, not the first half.
Yeah. Now that was. Let me see. Where am I going? We were having. They did do. We were answering Hogtie's question about what experimental work they had actually done. Did they actually, mutate organisms, or is this all been done using databases? And yes, they did mutate organisms. They used a plasmid to insert into a bacterium that didn't have the chromosomal gene, and so they could insert the plasmid with the various mutations that they were introducing into the gene that was carried on the plasmid and see if it restored function, if the organism could grow without exogenous biotin being added to the medium, or was it still an auxotroph? And so they actually did do some lab work there. But again, because of the way they designed the lab work, they're actually testing, can we get from B to C without an intermediate and without going all the way back to A. And I'm trying to look for the next thing I wanted as a screen share. Right, Cliff Jumper. See the one I gave you, which has got the grey box in?
Yep, got it.
That's their bunny for the next one, the grey box.
Yep.
This one.
I love this. I love this story. It's hilarious.
This one was quite interesting. Now this is prior to the 2011 paper. At this conference, the Wistar Retrospective Symposium, Anne Gauger had given a presentation And then she was asked by somebody to talk about some new experimental stuff. And so she gives the information. And she discusses leaky growth. And in those conditions, spontaneously arising was a novel variant which enhanced colony growth. Now, that's beneficial to the organism, enhanced colony growth, because it means that you are replicating faster, bigger colonies. And somebody said, so a beneficial mutation happened right in your lab. Now, bearing in mind that they're trying to point out that beneficial mutations aren't going to happen, and the moderator stepped in and ended the session. It's just that you couldn't make it up.
Exactly. But look at that. I mean, what would a scientist say to that? I mean, what would anyone being honest say? Yeah, obviously, because you know, natural selection, favorable diversity, and what are they? I mean, obviously, she was struggling. If the moderator stepped in, I wish I had been there. It would have been such a wonderful moment.
Oh, I know. And the thing is that if she's making a presentation saying that beneficial mutations cannot occur, and then gives an example of a beneficial mutation that popped up spontaneously under her nose, which she noticed and remarked on, and yet roll forward to 2011, and we have this paper which doesn't seem to be admitting that beneficial mutations could happen. And if we go back to the abstract of the paper, which is what I'm attempting to do here.
Let me point out that the Wistar retrospective was an ID conference to which regular biologists were invited, real biologists were invited.
Yeah, I'm going to get to next on that one.
Yeah, basically they were trying to do a little tap dancing and failed spectacularly.
Mm-hmm. Now, it's what is so impressive here is the sentence that Hogtie pointed out earlier. But evolutionary innovations requiring that many changes would be extraordinarily rare, becoming probable only on timescales much longer than the age of life on Earth. And that's what is in these.
Yeah, but bear in mind they think the age of life on Earth is about 6,000 odd years.
Well, they don't actually state that. That was something that I wondered about. But that was 2011 saying that, and yet in 2007 she'd admitted a beneficial mutation had popped up in her lab. Yes. Now back to the grey box one. The interesting thing about this is I'm going to. Let me see.
I was trying to remember where I put it.
And it was biotin again. Do you know, I've got this recollection that this biotin paper 2011 that we are looking at, I had it in my head that it was a 2007 paper. Has it been republished in 2011?
I want to say that 2007 they published on the last work on it's Barnase, which is what Doug Axe specializes in, and I think he was carrying data from wherever he was, King's College or something like that.
You see, here Anne Gauger responded to this, and here's what she said to this thing from Wistar. The work involves screening cells for their ability to grow on medium lacking biotin, an essential vitamin. Normally, bacterial cells can either take up biotin from their environment or make it themselves using a 4-step dedicated pathway. The cells I worked with lacked the first enzyme in that pathway called BioF, and thus could not make biotin. We were testing whether a structurally similar enzyme called KBL could be mutated enough to allow it to substitute for the missing BioF and thus allow cells to grow. Now that right there is describing the 2011 paper. Is it not?
Sorry. Oh, that's from the 2000s. Well, this is why I was asking before about where's this research being done. I mean, it's sort of standard amongst our group, that sort of those who kind of chuckle at creation or intelligent design or whatever, to say, well, they actually don't do their own research, right? Mm-hmm. It's all about sort of pointing your finger and saying, oh, this doesn't make sense to me, therefore God. And I've not heard any credible claims of doing actual research. That's why I asked the question. I kind of wondered.
No, they are. So there's a biologic.
They have labs and they have like gloves and goggles.
So Biologic Institute was founded and fully funded by Discovery Institute.
Yeah.
Biologic Institute. You can do the Google Street Map, and it's probably 2 rooms and an office. A couple of desks. So it's not a large facility. They actually have a quite large budget, and I don't know where that money is going to. And they've got a faculty of, I think, 5 or 6 PhDs. But apparently only Doug Axe and Ann Gauger have no other appointments for faculty. So my guess is the 2 of them sit there, and the rest of them kind of come and go. They've got an archaeology wing at the Biologic Institute, but they do have a lab.
Yeah. See, this is what's got me. So we accept that she's done this work in the lab, and she says the mutant strain I had found that could grow slowly on minimal medium could not grow on minimal medium plus streptavidin at all. Thus the mutation these cells carried, whatever it was, did not allow them to make biotin for themselves. It only allowed them to take up biotin from their environment more readily. This is the result I reported at the private meeting. No mistake, the mutated cells grew on medium without added biotin, but this ability was the result of increasing their ability to scavenge biotin from the medium, not because they could make biotin. Right? And this is why I'm thinking to myself, I had it in my head that this paper was 2007, and I'm surprised when I looked it up again, it was 2011. And this is part of the whole biotin thing that's been going on. If they've been working on it from 2007 to 2011, and they already know that they've seen this mutation occur, increasing the scavenging ability, so therefore they can grow. Then you could almost describe that as a scaffolding that allows the inactive form of it to mutate into something else. Because they've already got a spontaneous mutation that means that they can survive without the functioning gene. And that to me that says that what they're saying as the conclusion of the 2011 paper, they already know isn't quite spot on. You see what I'm getting at?
Again, it's like what I was saying before, it's that whole objective thing. They're not objective, they're trying to disprove something. They're just trying to replicate their confirmed bias.
Mm-hmm.
It's the opposite of science, right? It's starting with an outcome in mind and then arranging the data in such a way that it leads to that conclusion. It's fundamentally what's wrong with intelligent design.
There is a creationist mantra, which is, here's what I want to be true, and now I'm going to find the evidence to support that.
Yeah. And given that they already knew that this had happened in 2007, why do they not take that into account in the paper in 2011?
Do you know who the peer reviewers are?
I saw. Well, it says editor, but
Editor, yes.
That was who the one who did the paper, Michael Behe.
Do you know who the senior editor is for this publication?
Now, isn't it? It's either
it's not hard. It's the name on the top of the paper.
Axe.
Yes.
Yeah.
So Doug Axe is not only the author; he's also the head editor for this journal.
Yeah, and it's the, if you remember, the peers that they use for review are taken from the editorial board.
Yes. This is a newsletter. It's not a journal.
It's a newsletter. Although it looks great. This paper looks
it
so
does.
good.
It actually, have you seen the stylish paper? It's even more gorgeous.
Yeah.
Yeah.
I did want to say just one second. I did want to say when we touched on this before about, well, isn't it funny? Look at who the editor is and how they do their thing. That if you ever ask any of them about this, even sort of like YouTube tier creationists, one thing that we got to be mindful of is that they think it's fair when this sort of thing happens. So I'll make it as a parallel. But what happens when you tell a Fox News viewer that, oh, you must have learned that off Fox News? Well, don't you think Fox News is a little biased? I've never heard a Fox News fan ever say, no, I don't think Fox News is biased. I think they're the ones, the mainstream, they're the ones telling the truth. Their answer always is, well, of course Fox News is biased. We need something to balance out MSNBC. Like, the justification is because they have these views about these other things. Like, well, we have these other biased news sources, so we don't want a mainstream actual truth-telling news source. We want one that's biased the other way. And I've had this discussion where you point out, well, when they do quote-unquote peer review, look at who the peers are. I mean, they may as well just review their own papers. What do we need that for? That they will say, but that's the same thing the evolutionists do. Because when you submit a paper about some enzyme thing or fish sperm or whatever you're writing about, they don't ask people at the Discovery Institute to review your papers, right? They ask other evolutionists to do it. And that's sort of the common response I've heard. I think it's probably fair to address how that's not quite the same thing.
Isn't it also that there is a double peer review as well? As in, someone puts out a paper, someone reviews the paper, the peer reviews the paper, and then it goes out to the general scientific field, and then someone says, well, let's have a look at this paper, and then they try to prove it, disprove it, replicate it rather, not prove it. But they try to replicate it, and if they can't replicate it, they say, hang on, there must be something wrong here because we've gone through the system that you laid out, and we're not getting the same results. What's going wrong here?
No, I'm going to lean towards no, not really, as an answer to that. Perhaps because I misunderstand the description you're given. But remember that the research that would take place in whatever field would happen entirely within one laboratory or research center. And from that, there's an authorship pool that's drawn, and the paper is written up. And then it's a journal would be chosen, and then you would submit to the editor your manuscript and say, well, we think that this paper is suitable for your journal. We hope it is. And then the editor, sometimes with the help of the authors, will nominate a referee panel, and those referees can either do it or not do it. Usually you say, yeah, I'll review the damn papers. And as I've said before, so I won't repeat myself too much, but they don't actually review the data itself or try to replicate the experiment or do anything like that. They review it basically, is the paper of the appropriate format, is it, are the conclusions supported by the methods and the results that are provided in the paper. And you could have entirely forged all of it. You could not even done the experiment, just forged the whole thing, and they really wouldn't have a means of detecting it, except of course for the example that Concordance had given last week about one particular journal that does ask for your data. But for the most part, historically, that's not how it's done in science. And then once it's past that referee process, then there's changes to make or whatever else. They send you your proof prints, and the thing's printed. Right. The notion that scientists are seeking to replicate or to fail to replicate or find conditions under which this finding couldn't be replicated, that whole idea is post-publication, which is the lesson there being that when somebody asks you, like, show me even one, I dare you, show me even one peer-reviewed paper that proves evolution true. It's a stupid question, because there is no single peer-reviewed paper that proves evolution true. There is no single peer-reviewed paper that proves gravity is real, right? The peer-reviewed papers are to be interpreted as a set, because there are many examples of a paper passing peer review and then other people not being able to replicate it, and then that finding, even though it's past peer review, whoop-de-doo, here's your gold star, it just falls by the wayside over the next decade. That
or is
happens
retracted.
or even retracted, although that's
Yeah.
that's a fairly rare phenomenon. But
Yeah.
what I'm saying is the research is almost in its own way is somewhat of an evolutionary process, where you might find one lab that gets a couple of findings in a particular area, and then some other people might write a couple things that are related to that. They might get cited a couple times. But if it's not an interesting area or on attempts to replicate those findings, you don't get the same thing, and you can't sort of launch from that and generate new research. It's an area that ends up just dying off, even without retraction, those papers. So you can find examples in the literature of findings that are reported that aren't useful and are even just wrong and incorrect. And that's why when you do a proper literature review, you aren't looking for that one paper that proves this thing. You collect dozens of papers to see what's gone on in a very particular, narrow, small area.
Let me challenge back the first point you brought up, which is the equivalence between peer review in intelligent design within a single institute. The difference is the adversarial relationship in science, and I think that's fundamental to the difference between what they're trying to do, this sort of cargo cult science or anti-science, and what true science is. True science is adversarial. You've got people out there doing their very darndest to disprove what you have just asserted. Now it does, you're right, it comes post-publication, which is really frustrating, although it leads to these marvelous fights at scientific conferences. If you've never been to a scientific conference, it's one step removed from a wrestling match at times. But the difference here is that we've got people actively trying to poke holes in our research, and they are not. They're obviously ideologically uniform. Now, I can't prove that to a creationist. That's not something that they're going to accept from me. But if you and I are talking, I would say that is the fundamental difference between real science, real peer review, and the kind of newsletter approach that they're taking is, you know, it's a gauntlet, right? That's how the cute cartoons always depict it. You're running down that gauntlet, and your reviewers are supposed to be tearing you apart. You also mentioned that retractions are fairly rare, and I just happened to see an article today that 64 papers were retracted from Springer journals at the same time as a result of bypassing peer review. So maybe it's not so rare.
Yeah.
Well, I'd say historically they're not, and I recognize, and I think we all have to recognize that in the last years, yeah, we've had, and it's not going to be. I don't think it's going to be. I think yeah, it's sort of a blip now, but I think you're going to see more of it because I think most practicing scientists now are aware that it's well, we've always known it's not a perfect system.
Yeah.
But now we're seeing the just almost cancerous flaws in the system, really because of the internet, because of the different way that journals are distributed, the people's inability, like even practicing scientists sometimes can't recognize, a good article from a bad article, a good journal from a bad journal, and it's just that the whole publication industry has mushroomed in the last 15 to 20 years, really accelerating in the last 5 or 6 years, and yeah, and so now I think it's crackdown time. Now it's crackdown time, and I mean, okay, well, 64 out of how many?
Yeah.
It's a pretty small number, but it's
Tens
not going to
of thousands,
stop.
I would think, yeah.
Yeah, it's not going to stop. We're going to see some of the journals on particular tiers start saying, no, we're going to refine our methods. And it's not unprecedented. You remember, it was not that long ago. Now I think probably younger or older than any of us here, but there used to be a time that you sent the reviewers the typesetting manuscript, right? The same manuscript you would send for typesetting. Mm-hmm. And then the accusation, and it was a long, lingering accusation. Well, wait a minute. When you send that to, Professor Joe Blow at, Cincinnati, he knows who you are, and he may like you, or more likely doesn't like you. And these guys might, put that on the burner, right? So we went to this blind review system, so that you actually don't send the typesetting copy to those people. You send, the editor's copy, and then you send 3 blind review copies with masked information, and then when that's done, there's a totally different copy that's for typesetting, and there's a different reason for that. But I'm saying that there are sweeping changes to the review process that came sort of top down, where like the journals just said, no, we're tired of getting this accusation of unfairness, so we're going to change the very character of, now there are even things that you don't even tell them what university this is, because you could be, what the 4 of us could sit here and start our own research center and then send out blind review copies to these journals, and we may or may not get published because they no longer poo-poo articles that are written by people that aren't from the University of XXY.
I saw a journal just actually reading one today. And their practice, and this is one that I think needs to be instituted universally, is to put the names of the reviewers at the top of the paper. To me, that is such a critical thing, because then you've got reviewers with some skin in the game. One of the fundamental problems with the review process is that they're volunteers, and basically they're taking time out of their precious schedules for whatever reason to do reviews, and a lot of them are junior faculty with, like, they have to show that they do this as sort of like public service, and we want volunteers. There's no question, but the quality of review, I think, would improve if people had a stake in what they reviewed being published as is.
That does make a lot of sense. I mean, the difficulty is, how can you be sure whether the peer reviewer... I do like the idea that they're not paid, because if they're paid, then they could be bribed, so to speak. But also, how can you be sure whether this peer reviewer didn't say, oh, I haven't really got time to this, so I'm just going to skim read it and just tick it off?
I've done it. Slapdash. I've done
it. See, that's something that I've never done. I must admit, I've never done that. I've always sat and taken it to pieces. To actually read it, and I'm looking for the flaws. I'm not looking for the good bits, I'm looking for the bad bits.
I've gotten reminder letters. Have you ever let one sit for so long that the editor had to call you back and be like, TikTok?
No, I haven't, actually.
Well.
I'm a good girl, I am. But I think the difference between the way that peer review is working in the science journals and what we're calling peer review in biocomplexity, I think the difference is pretty obvious.
Oh yeah, and
I wouldn't mean that. I'm not saying that would mean that every single paper published there would be rubbish. I'm not saying that. But what I'm saying is that how much confidence can you have when you know that the editorial board are the ones who are also the reviewers?
Yeah, and I think that the discussion we're having here about, even my sort of candid admission, and it's funny to hear Concordance sort of say, yeah, me too. The peer review system in formal academics in the academy is like, it's not perfect, but there's an awareness of it, right? Like the members will recognize it. Like, we could be doing this better. There are many ways we should be doing this better. The discussion about whether there should be a separate pay for it. I mean, in a way. You are paid for it because you're given a professional services fee from your university. When you're in faculty, you're given a professional services fee from your university to perform a bag of professional services, and that includes the assessment of you're really not free to pass on all of them. You do have to put your oar in the water, because that's part of what you're paid for. But yeah, you don't get a fee, but that's been discussed. Maybe we should do it as a fee, and then maybe because whatever. I do like the idea of the accountability on the page and whatnot. But these are active discussions in academia of like, well, what are our goals here, and how do we achieve these goals, and are the methods to achieving these goals, do they change in the 21st century with this different type of technology? These are ongoing discussions, and there are journals that experiment with different ways of doing things. As Concordance is saying, let's put the reviewer's name on and see how that affects the quality. But our admission that it's an imperfect system isn't meant to sort of guide anybody to say, oh, so peer review isn't as good as people say it is. That sucks. Because, yeah, like you're just saying there, Fiona, I mean, it's an incredible void between the quality of peer review in academia versus what's going on over at Biocomplexity, where, oh, here I wrote a paper, what do you think? Oh, I should probably change the font on this, and we need more color in Figure 2.
Yeah. And it's worrying that the flaw in the experimental design wasn't caught. That's the sort of thing that should be caught by peer review.
You're talking about the cousin problem about the whole thing is predicated. I find that really interesting, and I want to say, just sort of turn our conversation a little bit if you don't mind, but I struggled with the paper. Like as I said, those 3 middle lines of the abstract, I knew I was up to the water was over my neck, over my chin, because I don't know enough about the details of enzymes. I mean, there's stuff that you learn, and then, like I learned within the first month of being out of school, that when it comes to organic chemistry and biopolymers, that it never came into play again. Now I understand, like U4U and concordance, this ended up being something that you embraced in your practices, where this is something you really need to dig in and learn more than I ever learned. But for me, it was just stuff you needed to know to pass exams, and it just never came up again. I forgot my point here. So I tried to understand this paper and the cousin problem on more of a macro scale. So one of the discussions I get into quite a bit online has to do with, well, can you prove that cats came from dogs or dogs came from cats or whatever? And you have to say, no, it's a common ancestor. Mm-hmm. And you can show them how you can trace back, because we have, the genetic similarity is one thing, but let's looking at the fossil remains. If you take modern dogs and go back and back and back, and you see see these slight changes as you go further and further back, you end up getting to the basal, the ancestor. Was it the Miacids? Is that the right name for it? I might have the wrong one. It's from the late Eocene. And then you do the same thing with cats. You go back and back and back and back, and you see them looking a little less cat-like, sort of more generalized cat. And then you get back to this sort of weird cat-dog looking thing, this very generalized sort of thing. And it turns out it's the same creature that hired the exact same piece of geography in the exact same period of prehistory. So it's obvious there was this sort of initial common ancestor that branched off into the feliform carnivores and the caniform carnivores. And the caniform carnivores split off into dogs and into bears, right? And then there's more splits as you go down the road. Now, here's the point. You all know that. Here's the point that I've been striving to make, and I sometimes struggle with, and I think it's a very important one, is that when they say, well, how can X turn into Y? How can A turn into B? And things like that. What we're forgetting is that basal historical thing, whether it's a amino acid sequence or an enzyme or in the example I'm using, a whole organism, a whole population of organisms, they're quite different because they're more generalized, right? They're very general in their function, and they may have a set of features. And then when you go from this basal ancestor down toward down the feliform route, there's going to be increased specialization as you go in that thing, and sometimes that means loss of function. And sometimes it means introduction of novel function or modification, very unusual novel modification of existing functions. And then, of course, down the caniform line, you're going to have the same thing, where it's increased specialization through loss of some other functions. And then some of those other functions will sort of increase in efficacy or sort of more highly specialized. So when you look at a modern cat and a modern dog, they're not just different from each other, but they're different in character from their very generalized basal ancestor, which is very sort of a generic sort of cat, dog, bear looking thing. Hey, we got thunder and lightning tonight. Awesome. My point here is when I look at this paper, I sort of thought the same thing about these enzymes. Is it not the case that when you get, like going back to your earlier tree, Fiona, where you started with A and then it branched out into the ancestor of whatever was A and D, or whatever, you remember that diagram, is it not the case that A may have been a very, very generic functional enzyme, and then when it's split off, those two groups specialize, including in some cases the loss of function? Now, why I think this is important is because once you've gone from A I forgot what our chart was, what our letter was, whatever, the basal one, and then you go off to, I think I've got the wrong letter, confusing people with the wrong letter, but whatever the basal one was, once you go off one branch and you're getting increased specialization, which may include a loss of function, to now say, well, we're going to go backwards in a way so that we can go from A to B or whatever the letters were, these two cousin groups. We're going to be able to do this conversion. You'd be talking about re-adding lost functions, which might not be an energetically favorable or environmentally guided process. And again, I don't know that I understand that well, which is why I'm trying to explain in part for my own benefit and in part for the audience's benefit. If you think about it like this cat, dog, bear problem, that cats are very specialized animals. And you look at the small cats with that weird cat eye pupil thing, right? That's a very, very unique feature. And to know, so well, how do we now convert a cat into a dog? And we're going to do it by going uphill on the evolutionary scale, and that now means they've got to regain round pupils and lose all of the specific cat things, and then start doing things towards the more dog-like thing. Or if you want a simian example, why is it that all the mammals have claws, except for like 5 extant species don't have claws? Humans, bonobo, chimps, gorillas, and the other one, orangutan, don't have claws. There's this one little subset that has fingernails instead of claws. Why? How does that come to be? Well, if you wanted to now say, well, how do you turn a human into a spider monkey? Well, we got to somehow work backwards and lose our flat fingernails and somehow regain claws, which is clearly something that was lost on the evolutionary pathway for simians. I don't know if those two examples help or confuse, but it's been on my mind, and that was what I wanted to get out.
Yeah. Usually when we talk about, I just want to say, like, when we talk about like losing features, I think the term that's usually used to describe that is evolutionary trade-offs. Like, sure, traits can be lost, but if there's really no use for them and they're certain in whatever environment there is, then it kind of evens out. Sometimes to actually gain new features, you have to abandon older features.
Yeah.
Well, the reason I wanted to raise that was to ask, too, if this was related to concordance, your earlier observation about, I think Fiona had used that word once, too, about this enzyme is broken, or this protein is broken. And I'm not really knowing what that phrase means by it being a broken protein. And then how this relates to the concept, or what people call the law of irreversibility. I think you're mentally a step ahead of me, because I just saw that link. Sorry.
Yeah, this is the Dallow's law. Go ahead.
No, you. No, or
okay.
Fiona. Somebody who knows.
No,
what
it's exactly. You're restating what Dallow observed in 1890. Now he didn't have the genetic understanding of the mechanisms involved, but he could simply observe that, yes, as you say, things go from. We have to be very careful, and this is why people close to the science have a very hard time communicating it. It's slightly pedantic to say more general to more specific. We don't imagine, a spherical mammal or something, that doesn't have any distinctive features. But you're right, adaptations that define that organism, which may have been present in the basal or ancestral population. There are a lot of different dogs that don't exist now, that you don't see the properties of dogs in wolves, right? So that's a fairly easy one to see. And we'll continue to have more specific types of terriers, and we'll continue to have more variations on the whippet. But you will continue to have divergence from a common ancestor. You'll never have convergence back to an ancestor, specifically for the reasons that we're talking about here, and that's what they tried to make it do. They basically verified Dallow's law of irreversibility of evolution, right? They stated that
with respect to enzymatic function. Okay,
yeah, absolutely.
That's what I was wondering if that applies to enzymes, because
Yeah, it's a bit like if you looked at a room. Oh, sorry, awooga, analogy alert. It's a bit like going into a room that is an absolute disaster zone and trying to put it back to the way it was. And if it has broken cups in it, you're in trouble because you can't unbreak the cups.
Is it because the second law of thermodynamics disproves evolution?
Oh dear God.
That's exactly it. Sure.
Well, hold on.
Oh, yeah.
If I can just say one thing, like on the comment of Dolo's law, is it possible maybe to have some reversibility in a population under a lab condition?
Yes.
Yeah, because
that's why it's not well in times. It's more of a concept than an actual law, because it is possible for the reemergence of ancestral traits due to changing selection. It does happen, but it requires more fitness, more fitness increase for that. It's a harder process than what we might say unadaptive change. The majority of evolution is more drift dominated. This has to be selection dominated for it to occur.
I've heard in some cases, usually when you're trying to treat viruses, obviously there are some variants of a virus that can obtain traits that make them resistant to possible cures or treatments. One of the therapeutic methods that I've heard that they do is that if someone's actually very sick, like with a certain virus or other disease, or really anything, sometimes treatment is withheld for a while, because what ends up happening is to have actually more of to actually get the antiviral properties, you'll have a lower population. So what ends up happening is the population, after putting them in a situation where they're not being hit, with or they're not being hit with treatments, the population will actually go back to a non-adaptive trait where they're no longer resistant to treatments anymore, because they don't really have the need to have that adaptation if they're not put in a situation where they need to adapt to it. If that makes sense.
Yeah. Yeah. It even happens in the wild too. You're talking about a manipulated fitness, where we're changing what is adaptive. They're all adaptive, but in this case, it's more adaptive not to have the resistance trait. But it also happens in the wild. There are documented cases where something reverts back to an ancestral form. But again, it's rare. That's really what Dalo would have predicted, is that of all the changes we see, the ones that move backwards along the ancestral line are much, much more rare. And again, we've confirmed that with this paper. I'm sorry, Fiona. Go ahead.
No, I was just going to say that it's slightly different if you consider bacteria and plasmids. There was a study done, I think it's 1979. It was a friend of mine who did it, and it was on antibiotic resistance in clinical isolates of Salmonella typhimurium. And they were looking at the various plasmids. And it was around about the time that the antibiotic resistance in this particular bug was on the upturn because of the use of antibiotics in cattle. And the plasmids that they had isolated from cattle, they were put into some of these clinical isolates to see what would happen. Now, they had 3 plasmids that they were interested in. 2 were quite small plasmids. Each one carried a different antibiotic resistance. And you could put the 2 plasmids into one bug, so it was then resistant to both antibiotics. But there was a 3rd one which carried chloramphenicol resistance. Sorry, I swallowed the wrong way. And it was a very large plasmid. And when they put the plasmid into the bug, in the presence of chloramphenicol, it was stably maintained along with the other 2 plasmids. As soon as they removed chloramphenicol from the medium, or they removed all the antibiotics from the medium, the chloramphenicol plasmid would disappear from the population, although the population would still contain the other 2 plasmids. And it was because of the bioburden that this large plasmid placed on the bacterium that it was only an advantage to carry the plasmid if the chloramphenicol was present. And if there was no chloramphenicol, then it made the bacterium so it was having to spend so many of its resources just maintaining that plasmid that it couldn't do it. And if one arose without that plasmid in it, it would then outcompete the ones that were still carrying the large plasmid, and so it would disappear from the population. And it's all to do with its population swings, it's the allele frequencies changing. It's like the rats in warfarin. In the presence of warfarin, you get warfarin-resistant rats. Take warfarin away, and you get warfarin-sensitive rats back.
But usually the payoff is, I think we've talked about this one before, the warfarin, the non-warfarin resistant rats tend to be bigger, they tend to have a higher fitness potential.
Yeah, but only when warfarin isn't present. And so the fitness of a certain phenotype is absolutely dependent on the environment. And what is good for one may not be good for another environment. And if the environment changes, and I suppose you could say that it can be forced along all sorts of paths. But when you're looking at the population level, then the backward steps can happen, but not all the way down a branch. That's not going to happen. It's to do with the allele frequency changing rather than the actual genetic variation in the first place. Does that make sense?
Yeah, I want to make sure we don't give the impression of adaptationists that we're not saying that all evolution is a result of adaptive change to a lot of it's just random. You lose an allele, you lose a rare variant, or you lose something that exists at 10% of the population has this one unusual trait. That may come to dominate the entire population or not, even if the environment has no favor for it. There's no advantage to it. It still may get lost. We call that fixation. It gets fixated. And there's a way you can time that out. Randomly, it either hits 0 or 1. And for a lot of changes, the ones that dominate the evolutionary history of an organism, it's those random 0 or 1 roll of the dice kind of changes that determine the cumulative effect of what that organism is or does or looks like. And those things, because they are random, you can't play the tape backwards, as Stephen Jay Gould tried to say. The idea of contingency, the accumulation of thousands of random events won't play back the same way a second time if you were to rewind the clock. And the same is true as if you're sort of forcing things back. Those random changes won't play out the same way a second time.
Well, some people might disagree with that. Some people might be philosophical determinists, and they might say, well, it would have been that way anyway, under any condition, because that's just how the laws of physics determined it to be. I don't necessarily agree with that view. I just wanted to make it clear that there are dissenting views.
Okay.
Those
people should go to Vegas, I think.
Yeah.
Right. I think we've covered most of the stuff about this paper and what the problems are with it. And why not being able to turn one cousin into another doesn't disprove that they share grandparents. I'd like to find a paper for the next episode, but that'll be in a fortnight's time. If anybody has any suggestions, let me know. It could be a paper that's a really good paper, and we can look at it and discuss the significance of it, or a paper which we find continually being used as evidence to support either intelligent design or creationism when it does nothing of the sort. I think that I'm interested in those ones because they're the ones that get trotted out all the time as, oh, this is peer reviewed and it's in favour of, or sorry, it's supportive of creation, or it's supportive of intelligent design.
What was that one that Trent Pearson was talking about when you had to explain to him Sergi and Wobble?
Oh, I can't remember. I've no idea.
That was from your famous, ill-fated Just a Chat. I still rub my temple over that.
That was not Just a Chat.
I don't have any idea about biology, but I got it.
Yeah, I don't think he had actually come with a particular paper.
No, he hadn't. No, I don't think so. I've never forgotten. I mean, third base wobble. I never want to hear it again.
The crazy thing about it is Lenny understood what that was all about. Georgina not so. She was busy thinking about something else. Lots probably.
Yeah. I mean, but I'm trying to think of any papers that stick out in my head.
Well, I have a quick question about this one, and it's sort of in general, and it's a bit of a tongue-in-cheek question. And I don't concordance with saying you had somewhat of a closing statement, but are we at the point now? So as we started reading this paper, one of the first things that caught our eye was the editorial board and look what journal it's in, and the eye rolling, and it took us a moment to get okay. Well, let's just actually look at the paper anyway. What the hell? Are we at the point or past the point now where when you see like Behe's name on an editorial board or Discovery Institute affiliated any organization, are we not just at the point now where it's just like, screw it, they're full of crap?
Yes,
I don't want to be accused of that, but yeah.
Actually, let me take this if I can, because here's where I think we missed the forest for the trees. The goal of the intelligent design movement and this publication and this newsletter / journal is to verify the positive signature of design. Mm-hmm. And I think not addressing the question, if this is true, if we exclude the methodological flaws, is this truly evidence for design? And I want to really emphasize the idea of a top-down design. Mm-hmm. I don't think I talk about this enough when I talk about intelligent design. They're taking existing enzymes and seeing if they can achieve new functions or fix functions that are already broken. That's not design. What's interesting is that they seem to abandon the whole idea of proving sort of a top-down approach. If you were walking in the woods, and we're not going to do a watch because that's stupid, but let's say you see a jumble of rocks that look like they would be a good place to sit down or look vaguely like a throne, mm-hmm. right? That would be very different from walking in the woods and seeing something that was carved marble. They seem to be looking for jumbles of stones that you could sit down on, which can be explained by natural processes. They don't even seem to be setting out to prove the positive signs of top-down design. Someone with an intent in mind, creating something de novo, novel, creating from nothing, mm-hmm. A protein to do a specific function. Does that strike you guys as a little odd that they're modifying one thing to do a different job? They're taking junk and trying to find new functions for it? That's a tinkerer, not a designer.
Yeah, that has struck me. And what has also struck me is that they keep the phrase I keep hearing is, when you say, what do you mean evidence for design? And the only answer I get for that is it looks designed. I want to know what something that wasn't designed is going to look like. What's the negative control for the experiment?
Yeah, I know what you mean. It reminds me of when I was dealing with the 9/11 truther types. And, you get people saying, when the planes hit the buildings, that doesn't look like, that looks CGI. That doesn't look like what a plane looks like hitting a building. It's like, well, what exactly does a plane look like hitting a building? Because we've only ever seen it in movies. Yeah. Until that day, you just didn't see these things. So what exactly does it look like?
Yes, that I think really gets to the heart of it. If you want to design an experiment to test for design, what is going to be your negative control? You can't carry out any experiment unless you have a negative control in there and a positive control. And our problem is this: the one thing that is unique to life is that it reproduces imperfectly. So what is our positive control going to be? Either you're going to have to say, well, we need to use life, in which case it's exactly the same as a test. Or you're going to have to use something like a car, which isn't a biological system, which doesn't reproduce imperfectly. So you have no positive control. You have no negative control. How are you going to test for design?
My very first video on YouTube was a challenge to creationists to come up with a gene that was not homologous to another gene, that showed no evolutionary relationship to another gene. It doesn't sound that difficult. We established a couple of criteria, a certain degree of homology, doesn't match up with anything in GenBank, and a number of things were advanced. They came up with genes they thought were unique in humans, and of course you find the chimp homolog. It's quite simple, and invariably you find a homolog. They could have even looked for orphans, and spelled ORF as an open reading frame, orphans, which are supposed to be genes that don't belong to a family. But that's only because people haven't done the work to draw the evolutionary trees to figure out where they fit when you run them as a search of, does this DNA match any DNA anywhere in any database? The answer is invariably yes. So if there is a designer, and I don't think any of these experiments were really designed to test that in any serious manner, if there is a designer, it's a tinkerer. And that's kind of how we know nature works. It takes what exists and tinkers with it, finds new ways to use the old tool, makes a copy of the tool, and then it modifies it a little bit, and hey, what do you know? This does a new job that I want to do. And that's, again, I almost feel like this was a confirmation of that principle in this Axe and Gallagher paper.
And they didn't, but they didn't see that. And I mean, I've been guilty in the past of being so blinkered, or such tunnel vision in what I'm doing, that I've designed an experiment, and I've gone away and I've come back and I've looked at what I designed and I thought, oh, good grief, Fiona. Because of the tunnel vision, I didn't see the hole in it that you could drive a truck through. And in fact, when I was doing my PhD, I did that. And I actually did the experiment and all the rest of it. And it was only when I came to the write-up, I realised the huge hole. And it was actually interesting because of the flaw in the design, it meant that the experiment asked a different question to the one I thought I was asking. And it was actually a more interesting question, and also answered that more interesting question. But that was more through luck than judgment. I mean, it meant a complete rethink of some of the writing I was doing. But it was, I couldn't believe how daft I'd been in the flaw that I put into the paper. Into the experiment. But it was because I was focused so narrowly on it. And I see what they've done with this paper. It's very like what I did with that experiment, and I didn't see the hole in it until I came to write up. And I was far enough removed from the experiment itself that I no longer had the tunnel vision. Now, I believe, I have read in a few places that Gallagher has admitted that the choice of enzymes was not a good one because they were far too far apart. And they should have chosen enzymes that were much more closely related. So that would mean that she's admitting there is a flaw in that study anyway, and that what they were attempting to do was very, very unlikely to happen because it was too big a gap. but they haven't repeated it, as far as I'm aware, with more closely related enzymes.
They don't need to,
Yeah.
The creationists have got what they want. They've got a paper they can cite and say, look, here's a peer-reviewed paper. It's got nice colored graphs, and it says this, and the subtlety of it. They're going to miss the fundamental logical mistake. I mean, again, maybe we should have addressed this sooner, or we did. I don't know, but the basic logic here is, well, if evolution is true, it should be possible to go from A to B in the laboratory. Premise 2, we couldn't go from A to B in the laboratory. Conclusion, therefore, evolution is false. Right. Well, you know what? I could do the same thing. I could say, well, if evolution is true, I should be able to, convert whatever enzyme, egg yolk. Actually, I have some eggs in my fridge. I could get the protein, egg albumin, the egg white protein, and if I can't convert that to monkey sperm, then evolution's false. Right? And what have I got? I got a whisk and some butter, and I don't know. I'll let you know next week whether or not I was able to do it. And if I can't, well, then evolution must be false. And therefore, my God is true.
Which is that whole claiming Mount Improbable thing, isn't it?
Mm-hmm.
Yeah. Does anybody else have anything they would like to add here? Apart from the odd head, desk, and face palm. We all done then? Right. So we'll need to think up another paper for a fortnight's time. I'd appreciate it if you could all turn up again for a fortnight's time, if it's possible. And I'd like, as I said, I'd like to stick to those papers that get used by the design people or the creationists when they've misunderstood the paper, and actually look at the paper, or when they're using a paper that is just a load of marsh gas. So any suggestions, please let me know. And if you let Becky know as well. If it's going to be difficult to get a hold of the actual paper, if whoever can get a hold of it could pass it round us all in full text. And be back in a fortnight, hopefully. And I'm going to end the broadcast now, so say bye-bye, everybody.
Bye-bye, everybody.
Bye, everybody.